And now we go to Hanadie Yousef, who will speak about harnessing the regenerative secretome of human embryonic stem cells to rejuvenate aging tissues. Please.
Great, thank you. I just want to quickly start by thanking the organizers of the seminar for inviting me and Irina Conboy for recommending me. My name is Hanadie. I have been the CEO and co-founder of Juvena for the last 3 years. Prior to that, I received my PhD in the labs of Professors Irina Conboy and Dave Schaefer at Berkeley, followed by a 5-year postdoc at Stanford. And I'm excited to share in the next 15 minutes our story about harnessing the regenerative secretome of hESCs to rejuvenate aged tissues. And so at Juvena, it's our mission to develop novel breakthrough protein therapeutics for degenerative diseases caused by both aging, as well as in many cases accelerated by rare genetic mutations, by mining the regenerative secretome of hESCs using state-of-the-art proteomics and machine learning. Now, what really prompted me to co-found Juvena was just this burning question that we all have at this seminar in, how can we actually promote healthy aging? We all know that we're really facing a chronic epidemic of unhealthy aging. I mean, even myself looking at, my parents and aunts and uncles, if myself and my relatives don't intervene, both in lifestyle interventions as well as therapeutic interventions, it's almost inevitable that once we reach 40s, 50s, and beyond, we're going to get diabetes, obesity, cardiovascular disease, stroke, vascular dementia, cancer, right? And so more than lifespan, what I really care about is how can we promote healthspan? How can we slow down the degenerative process? And for folks that, didn't have the opportunity to intervene at an earlier stage, even if you already have chronic symptoms, can we reverse that degeneration and bring you back to a healthy state to live out those later decades of your life in happiness and with high energy? And so what we really turned to at Juvena was some key discoveries and observations made over a decade ago during my graduate studies in the Conboy lab, and then really further exemplified and studied at Juvena on really turning to the most pro-regenerative source on planet Earth for human beings, which are proteins that are secreted by human embryonic stem cells, which we know hESCs can develop every tissue in the body. And what myself and colleagues observed is that by purifying a subfraction of the protein secreted by human embryonic stem cells and then adding them onto aged and degenerating tissues, both mouse and human tissues, we could actually promote regeneration, whether it's enhancing acute injury, muscle regeneration in an acute injury model, or promoting aged chondrocyte precursor proliferation and viability from chondrocytes isolated from osteoarthritic knees, or even promoting the survival of cortical neurons in the face of A-beta. We published on this over a decade ago and have now several issued patents, both out of Berkeley and Juvena, really showing and demonstrating that regenerative potential. And so we wanted to turn this into drugs for people to reverse and slow down the aging process. But of course, it's unfeasible to take a cocktail of what could be thousands of unknown proteins and just inject them into people. It could cause cancer. It would never be approved by the FDA. And so what we really set out to do at Juvena was establish a systematic method for identifying proteins in the secretome that could be master regulators or key drivers of the regenerative process for a particular tissue, and then go through that preclinical validation and clinical translation and FDA approval to turn it into a drug that we can distribute to millions, if not billions of people. And so to do that, the first thing we did was really perform quite extensive quantitative mass spec-based proteomic analysis, comparing and understanding the secretome of the subfraction of proteins in hESCs that are pro-regenerative compared to, for example, differentiated cells or hESCs going through the differentiation process that lose that regenerative potential over time. And it turns out that we have about 1,000 bioactive human signaling proteins in Juvena's now proprietary regenerative library that we're mining to identify proteins, either individually or in combinations of 2 to 3, that can really drive the regenerative process for a particular tissue, rejuvenate stem cell function, promote regeneration, restore tissue homeostasis, to really advance healthspan. And once we developed this protein library, we then set out at Juvena to really build a very efficient identification, high throughput screening, and preclinical development platform to rapidly bring these protein therapeutics to market quickly and efficiently for diseases that the FDA considers diseases that ultimately are caused by the age-related degenerative process. And to do so, we have built a machine learning enhanced platform that really combines image-based, robotics-enabled high throughput screening of primary human stem cells that are isolated from age and degenerating tissues, as well as human in vitro disease models, combined with multi-omics modalities, namely proteomics and transcriptomics, with machine learning in order to identify and therapeutically screen regenerative proteins that we can then turn into drugs. Ultimately, as we build this platform, we really want to create a map of regenerative protein biology that we can use as an engine to really bring multiple therapies to market quickly and efficiently. And so to quickly kind of go through how we do this, of course, I mentioned we developed our library. We also now have a system and a machine learning predictive model that also takes advantage, of course, of our team's really domain expertise and understanding of the mechanisms underlying the dysregulation of signaling that occurs in specific tissue types to rank order proteins in our library, and then screen them individually in synergistic combinations of 2 to 3 for their ability to enhance and reverse a particular phenotype that's representative of how the disease progresses in a given tissue, such as muscle in humans. And so even in the high throughput screening process, we've also developed several deep learning and in the preclinical process, models that really can accelerate our ability to quantitate that regenerative effect. Once we have hits in vitro in mouse and then human cell lines and primary cell models, we then very extensively validate these protein therapeutics, these human signaling proteins, for the ability after injection to rejuvenate aged tissues using a variety of preclinical animal models, from which I think is the most potent natural aging through to specific disease models that are also indicative of the degenerative process. Once we do that and we identify key hits in vivo, we can then engineer these proteins to really have be better versions of themselves, have better pharmacokinetics, really turn them into drugs that we can inject subcutaneously, just like insulin, right? It is used and administered in people to really enhance tissue homeostasis, promote tissue regeneration and repair. And so as mentioned, we've developed several kind of machine learning techniques integrated into this high throughput screening and identification and preclinical process to really rapidly bring these therapeutics to market. And so we, very quickly, I'll just mention that we've built a semi-supervised deep generative model using both proteomics, the immense scientific literature on our candidates, as well as the structural features, the quantitative structure activity relationships, to identify and predict and rank order the proteins in our regenerative library for their ability to likely enhance tissue regeneration. We then have developed several deep learning based models that really accelerate our high throughput screening and preclinical quantitation and validation. And what's beautiful about really building and growing a machine learning engine to bring this to market is that our Juvenet toolkit is getting just smarter as we go, right? Where we've developed these prototypes, these models, and then we can consistently, as we get our data and our own training data that we label and through every round of validation, make the model smarter and work more efficiently and have a greater likelihood of identifying proteins that really work potently and efficaciously. And so in the last 3 years in building this, we've not only established our library, but validated several therapeutic proteins in both mouse and human cell models. Once we had that hit of now over 11 proteins that really work potently to enhance specifically muscle myogenesis and muscle regeneration, we then validated these in several preclinical animal models with the ultimate goal of finding key proteins that can activate and signal through core regenerative growth and survival pathways that can reverse the aging process, promote tissue regeneration and health span, and be used to treat a variety of, in this case, muscle wasting diseases. And so to just kind of show quickly an example of that, here is our current pipeline. And I should mention that, we have developed and identified and discovered several combinations of 2 to 3 proteins, which I think are the best in really mimicking what the original subfraction of the hESC secretome does. But to raise money from investors and really also get this to market as quickly as possible, we've then identified in these the best protein therapeutic in this library, in our validated results, that even on its own has a good enough and potent enough effect that we can bring that to market first. And so I'm going to give an example of this therapeutic. Of course, I can't share the identity yet, but I will in the near future, of JUV161. And so, JUV161, when we added onto several primary human muscle precursor cells derived from donors ranging from 68 to 75 years of age, we can very potently activate myogenic transcription factors, growth and survival signaling to promote better fusion. And we know that with the aging process, as well as improved survival of these precursor cells. And we know that in aging muscle, one of the key hallmarks is this decline in the regenerative process, inability of muscle precursors to become activated, to proliferate and divide sufficiently, repopulate an area of injury, and then form new muscle fibers. And so this potently works to do that. When we inject this protein, of course, that model really reflects acute injury. So that was the first thing we very quickly did and showed, in fact, that this protein, when injected acutely into injured mice, can enhance regeneration significantly, nearly to levels of young, healthy mice. But then we really wanted to know, well, can we take this protein and inject it systemically through subcutaneous administration of functional output of even mature fibers and improve the existing muscle structure and function to promote repair, regeneration, and lead to improvement in treadmill performance and grip strength and endurance and distance run. And of course, we also really tested it chronically to assess safety and toxicity, and it was safe and non-toxic. And so this is just, highlighted a few examples of the data we got in our multiple validated animal models with JUV161, where it can enhance grip strength and distance run in a treadmill, promote grip strength, and also just increase muscle fiber size in a glucocorticoid-induced muscle atrophy model, and enhance regeneration nearly to young levels in an acute aged injury model. The dotted lines are here representative of how young healthy mice perform in these assays. And going on the concept of and the kind of theme of this seminar, I do truly think that out of all the models we've tested, the best indicator of if a human protein therapeutic will ultimately work in humans is trying to look at natural aging and evolutionarily conserved mechanisms of aging, from mice through to humans. And so it's that, natural aging model, which of course in every single model that we've developed or established, I should say, at Juvena, we've done extensive validation of that model to do a thorough power analysis and really make sure that the results we're getting are, true and consistent and quite believable. And so, just as an example, in the age of mice we use, we choose mice that are 21 months of age through or older, but it really, you need them to get at least that old to really start seeing this clear decline in, functional outputs of muscle function and health, such as grip strength and time to exhaustion. But nevertheless, there is variability. And so you really got to be careful and ensure that you're, using multiple models, using enough mice to really validate those results. Ultimately, we are developing a strategy at Juvena where we can kind of, hop indications, right? In my experience, it's very tough to get someone to fund us going to market first as a small company for just, a huge unmet need like sarcopenia, which ultimately will affect everyone as we age, right? Age-related muscle wasting. But what's beautiful about our approach is that we're identifying factors that we can turn into drugs that can really target core mechanisms of degeneration and be applied to a variety of muscle wasting and age-related diseases, ranging from the normal aging process, but also from, genetic diseases such as dystrophies, where a rare genetic mutation many ways causes this accelerated aging phenotype and degeneration, and we're seeing efficacious results there as well. And so the ultimate goal will be able to, get approval for a rare genetic disease, but then be able to really expand to be able to treat a variety of muscle wasting diseases, which are critical, right? Given the state of just our aging population and the fact that there's few, if any, FDA-approved treatments on the market today for some of these core causes of unhealthy aging and chronic degeneration that we're experiencing as a population. And so ultimately, too, our library of therapeutic candidates and platform, will be used and expanded so that we can identify tissue-specific therapeutics that can promote regeneration and promote health span across the body, right? Whether it's targeting neurodegenerative diseases, osteopathies, cardiovascular, or our current focus in multiple myopathies. With that, I'd like to thank you for your attention and time, and I'd be happy to answer questions either in the chat or if there's time right now. Thank you so much.
Yes, there's question. There's the time for one question.
I have a question.
Please.
Great.
You say that have you ever tried injecting the entire secretome into your mouse models of the stem cells, the embryonic stem cells?
Yes, but in that model we use an acute injury model because it's actually quite laborious to really culture hESCs and then try to produce and purify enough for, let's say, a systemic administration. And standard wet labs without having these facilities that we're just not able to have at this time. But then that's also the beauty of our approach is once we know what the actual sequence is and some key proteins in the cocktail that can act on their own to promote regeneration, we can then just use CHO cells, right, or 293Ts, for example, to recombinantly produce them and then even engineer them to be more like therapeutic drugs with better pharmacokinetic properties.
Thank you very much.